Your book has been a great inspiration for this theme we are addressing this year, which is the theme of long duration, la longue durée, as Fernand Braudel called it. And in a way, it's the question, of course, also given the ecological crisis, the extinction crisis, how to basically liberate the world out of this short-termism, which is so destructive, and think about more long durational pattern. And that, of course, brings us right away to your extraordinary book, The Entangled Life, and how fungi actually make our world, change our minds, shape our futures. Because you talk about these extraordinary symbiotic systems, really, of fungi and plants. And you talk about some of these tissues, actually, which are very enduring. Some of the mycelium tissues are clearly extremely long durational structures. So I wanted to ask you to tell us a little bit about the book, about the Entangled Life, and how you see it connected to the theme of the long duration.
There's lots of ways it could go. So yeah, fungi are they're a good subject to put in this theme because they've been around for over 1 billion years of evolution. So they've had a long history on the planet. And one of the interesting things about fungal evolution is that when you look at, say, the fossil record and you find all these different animals that have gone extinct, you see all sorts of forms. You see the most astonishing variety and diversity of animal forms. But when you look at the fossil record and you find these very ancient fungal specimens from hundreds of millions of years ago, they look pretty much just like they look today. And so fungi live most of their lives as mycelium, not as mushrooms. Mushrooms are just the fruiting bodies of fungi. So most fungi live as the branching, fusing networks of tubular cells, which is how they feed. And when you see fossilized mycelium or fossilized other fungal structures, they look very similar to the way they look today. And it seems though they hit on some kind of near optimal way of being very early on in their history, and haven't had to do a lot of change since then. Unlike animals, who are always inventing new ways to find food and put it in their bodies. So this is one way of thinking about the long history of fungi. But another way of thinking about the long history of fungi is in the way that they form relationships with other organisms. So their relationships with algae, for example, which became what we think of as land plants and gave rise to everything we know about the terrestrial biosphere, or at least gave rise to the terrestrial biosphere as we know it. This is a relationship that lasted hundreds of millions of years, and right at the very beginning, when algae were coming onto the shores of lakes and rivers, and faced with all sorts of new challenges that life on land brought with it, they formed these relationships with fungi, and the fungi were able to scavenge in the ground and find nutrients and water, and to exchange that with their plant partners for sugars and lipids, energy-rich carbon compounds that the algae or the plants make in photosynthesis. So when we think about these long relationships and the way that fungi have shaped so much of life as we know it, creating the conditions for animal life on land, including our own, we can see these long imprints of these dynamic relationships. And I think that's a really helpful way to think about these long histories of life, is in terms of these long relationships in which both partners shape each other and are shaped by the other, to the point where you can't actually think easily of one without thinking of the other. It's a bit like those drawing hands, that Escher painting, where you couldn't have one hand without the other. So this is one way that I think about the long history of fungi.
Beautiful. And of course, it also is important what we can learn from fungi in terms of the environmental crisis, the extinction crisis. And I've been actually reading this weekend, rereading the amazing book Dub by Alexis Pauline Gumbs, which is very much thinking with Sylvia Winter. And of course, Sylvia Winter is someone who is studying the multiple histories of knowledge. And it made me think of you, because I think in a way you've been studying many different histories of fungi in this book. It's not just one fungal history. It's a lot of different histories of fungi, and I wanted to ask you about that. And also, I mean, Alexis Pauling-Gams writes and says that we have the opportunity now as a species fully in touch with each other, and of course that refers to technology also, to unlearn and relearn our own pattern of thinking and storytelling in a way that allows us to be actually in communion with our environment, as opposed to a dominating colonialist separation from the environment. So the question is also, how can fungi help us to achieve that?
Mm-hmm. Yeah, I found this book very powerful as well, Dove, the one you mentioned. And so I think there are lots of ways that fungi can help us to overcome this separation, or rather to overcome this story of separation, because we can never really be separate from the systems that sustain us. We can make stories that cast ourselves as separate. We can make stories that give us neat, defendable borders that separate us from other humans and from the rest of the living world. But this is just a fiction. So I think fungi can really help us to break down these fictions, these unhelpful fictions of separation, in lots of ways. I mean, one way is just in learning about them, in thinking about them, in finding out how these organisms live their life, which pays so little respect for neat edges and borders. For example, one fungal network, you can take a fragment of this network and it can regenerate an entirely new network, an entirely new organism. So where does one individual start and stop? Two networks can fuse with each other to form one large network. These organisms interpenetrate other organisms to the point where it's very hard to say, as I was saying before, where one organism begins and the other ends. If you take away this organism, the other one dies because it no longer has this crucial life support relationship. Then to what extent can you think about this organism as separable or separate from the other? And so I like very much this piece of writing by Jack Forbes, and he says, you can cut off my arm and I'll survive, cut off both my arms, cut off my legs and I'll survive, cut off my ears, take out my eyes and I'll survive, cut off my nose and I'll survive, but to get rid of the air I breathe and I'll die, get rid of the water I drink and I'll die, And so we think of these symbiotic relationships, that's only the beginning. These are the other organisms that we depend on and who depend on us. But there's also these, what we don't normally think of them as living systems, but the air and the water, which we are completely continuous with. We are porous bodies, we are porous events. And so I think fungi can help us remember this because their lifestyles are so flagrantly uncontained by our flimsy narratives of separation. There are other more literal levels as well, of course. Fungi form, psychedelics is one example when people have experience of, say, psilocybin or other psychedelics, many produced by fungi, then a common experience is a sense of losing track of where we stop and our environment begins, sometimes described as mystical experiences, a sense of losing track of one's sense of self, and or a sense of losing track of oneself, rather. And so on a very literal level, we can have our boundaries and our categories confused by these organisms and the chemicals they produce. And so I think there are a lot of ways fungi can help us to overcome these tired stories, which really I don't think serve us right now.
So learning from fungi in many different ways. Of course, part of your book, and I think a very important part of your book, is also to make the invisible visible in a way. And I grew up in Switzerland, and so one of the first artists I came in contact with was Paul Klee, the work of Paul Klee. And Paul Klee, it was always the definition of art, Paul Klee gave, that art makes the invisible visible. It's interesting, of course, because we can also think of Italo Calvino's in Invisible Cities, but in your case, it's the invisible mushrooms, it's the invisible fungi. And you're right that they are everywhere. They sustain us, our lives, as you explained. They are inside us. They change the way life happens. I mean, you explained that very beautifully in the recent fish at the Serpentine, and when you were in conversation with Lucia Pietrojusti and Felipe Ramos, that really a majority of fungi are undocumented. I think you said 90%. And that, of course, leads us, and that was the subtitle actually of the Serpentine Conference, which you inspired very strongly, leads us to this idea of the understory of the understory. Can you tell us a little bit about this invisible world and how to make it visible?
It's such an important question. Lynn Margulis had this great term. She described these worlds as subvisible rather than invisible, because which I like, because the invisible suggests that in principle they're not visible, and it rather sets it up to fail. But subvisible just sets it up with a challenge. And one of the exciting things, and I think one of the reasons why fungi and other microbes are having so much attention right now, long overdue attention, is because of these new technologies that allow us to make visible, to see, or to otherwise pay attention to these realms of life which take place on such small scales and time scales as well, which are very difficult for us to notice with our own unaided senses. So part of the challenge is technological, and I spend time using different microscopes to try and render these fungal relationships with roots more visible, and something that we can get a feel for, some kind of intuition for, rather than just have them flat. I like making these root scans, which you can see in 3 dimensions, and I'm working with a friend who's found a way to put them into VR space, so you'll be able to navigate them even more intuitively. I always like the idea of printing them out, huge, life-size, and in kind of silicon, so people can clamber around inside these fungal networks in 3 dimensions and have a sense of the intimacy of the associations between these roots and these fungi. But in the book, obviously, this was not how it was happening, and it was much more in words. And I think there's a lot we can do with words and story and metaphor to explore microbial life. And I think music is also very helpful. And one idea I put forward in the book is that polyphonic music can help us to understand the lives of these organisms, which are so distributed. There are so many hyphal tips of one part of a fungal network, all growing at once, all making decisions at once, living in this very parallel way. And I think there's analogies with polyphonic music, where you have lots of different melodies all happening at the same time. Each of these melodies would stand alone as a melody, but when they're all together, they combine into something else. They kind of fuse into this larger whole. And when you listen to them, you can follow 1 or 2 of the melodies with your mind, and then jump to a different melody and follow that with your mind. Or you can kind of lean back and hear all of the sounds at once and listen to the whole that they create. And so I think actually polyphonic music is an astonishingly effective way to explore ecological simultaneity and the ongoing eventness of ecological processes, and one that we don't normally think of as an aid to scientific understanding. We normally think of it as locked up in the art world, or just something that people can appreciate as pretty music. But I think there's a lot of power there, and there's a lot of potential to explore polyphonic sound and use this as a way to deepen our understanding of our chemical processes and biological processes in the soil.
Thank you so much. And I'm really glad that you bring the conversation to music, because my next question has exactly to do with that, because you talk about the connection to composing, but also decomposing. And without the decomposing, there couldn't be composing. And you mentioned this story, which is really the beginning of the book, which is an experiment your father made actually when you were a kid, with a bottle. And I was very fascinated by that, because more or less, I mean, I suppose in the 90s, when you grew up, we did this experiment where we invited your father to do a telepathy experiment at the Musée d'Art Moderne de la Ville de Paris. I lived in Paris at the time. And Rupert, your father, came to Paris, and it was an amazing experience to do this experiment with him and learn from him about this sort of very DIY way, how he approaches experiments. And so then when I read that you had your epiphany really for your entire work with such an experiment with Rupert. I thought it would be great that you tell us that story and the importance out of that really of you understanding the connection between composing and the importance of decomposing.
Mm-hmm. Yeah, my father's really good at these DIY experiments, experiments on a shoestring budget, which I really appreciate because there's no reason why experiments have to be expensive. We exclude ourselves from scientific practice by assuming that it needs to happen in a lab with special equipment, but actually you can do a lot in your kitchen or in your garden. So so the experiment was, it was a very simple one. I'd been thinking about, I used to make these piles of leaves in the garden, and I was lying these piles of leaves, and I loved the smells and I loved the atmosphere in them, and I could hide in them, and they were exciting places for me to spend time. But these piles of leaves would decompose over time. They would disappear. They would sink into the soil, it would seem. And so I remember asking my father about how this happened and what was this process. And he talked about fungi and these other microbes that decomposed the living world, these dead parts of the living world. And I struggle to understand this because, of course, how can you understand this thing which is invisible? These organisms are invisible, and they take it to the or subvisible, and these processes happen on a timescale that make it hard for you to actually see it happening. You can see the consequences of it over time, but it's hard to relate directly. So the experiment he proposed was a very simple one, where we got a clear plastic bottle and cut off the top, and we filled it with layers of sand and soil and dead leaves. And so you could see through the walls these very clear bands. And then we really put some earthworms inside. And over time, you could see, and they were not very long, actually, over days, you could see the earthworms moving between the layers, carrying the different layers into each other. And the sand you could see in the soil, and the soil you could see in the sand, and the leaves you could see in the soil and the sand. And it created a kind of marbling effect. And it was a beautiful way to see how mixing happens in the natural world. And so he said, well, look, these worms do this mixing, this stirring. But there are many other creatures that are like worms, but much smaller than worms, that aren't worms. This is what these microbes do. It's analogous to what these worms are doing. And so this really helped me to understand this process and to think about these decomposers as an essential part of the living world. We live and breathe in the space that decomposition leaves behind. We so often take it for granted. We live in this negative space left behind by the action of decomposers. And so composers, everything is just making a life, creating a body, building a plant, building whatever it's doing. It can only do that if decomposers have, first of all, decomposed and recycled this once living material. So the composing and decomposing are just, again, the Escher hands, these key interlocked processes, but about which we think too little, I think.
Now, you also are very much into the DIY experiment, and so am I. I started in the 90s an exhibition called Do It. Actually, Rupert participated in that quite at the beginning. It's an exhibition where we have recipes and instructions and how-to manuals by artists and scientists and architects, and then people can just do it. And it was actually fascinating during the several lockdowns that this project, because there are many different versions out there, translations, books, was revisited a lot because people kind of had a desire and a necessity, I think, to go beyond the two-dimensionality of the Zoom, of the screen, and do things, right? And so all of a sudden, people started, I got lots of alerts of people who would do projects out of these Do It books and realize these recipes, and so Mare gives autoprogettazione, he gives a instruction actually how to transfer the knowledge, how to make design, and one can do it oneself. Or Louise Bourgeois tells us how to smile at someone, and at the same time we have Franz West who tells us how we can transform the broom into a sculpture. And you also work a lot with such DIY moments. I was amazed by your do-it-yourself video, which went viral actually at the moment of the book launch, where you somehow give us instructions how we can actually eat the book. So I was wondering if you can tell us a little bit about that. And then also, because it's very important for us with E.A.T that the engadinarttalks are not just a Zoom screening. It's very much, as Beecher Kuriger says, it's very slow TV, so there are going to be long durational moments of slow TV, but it's also moments of participation. So I was wondering, after maybe describing to us how one could eat your book, if you can maybe tell us also something else which maybe we can all do at home related to fungi.
Mm-hmm. Yeah, good ideas. Well, the eating of the book, it came about because I think it's very easy when, especially in the sciences, when we're talking about the living world, to abstract ourselves from it, to imagine ourselves as separate from it, especially, and I'm sitting in front of a screen for so long, pushing these words around on a page, talking about these rich lived processes. And I could feel the abstraction setting in. And so I wanted to have an antidote for that. I wanted to fold myself and fold the book back into the living world, back into these metabolic processes that I've been discussing. And I thought the best way to do it would be to feed the book to fungus, and then let the fungus sprout mushrooms, and then eat the mushrooms myself. And then there's absolutely no way that I could sit back with my view from everywhere and nowhere, like it's so easy to do. So it was a very good medicine for that, a very good medicine for my human-centeredness and my view from nowhere. But it was really good fun too, because it allowed me to eat these words that I'd written, and it allowed the fungi to also to eat the words, and it felt only fair to do it like that. And my brother Cosmo had this good idea that he said, well, how are we going to know what the fungi are making of it as they're eating the book? So we sent a copy or two along to a sound ecologist called Michael Prime, who rigged it up with electrodes so that we could hear the electrodes translate the bioelectrical activity of the fungus as it's eating the book into a sound. So you can kind of eavesdrop on the fungus as it's digesting the book. And so that gave us a lot of enjoyment to be able to listen to what the fungus was making of the words about the fungus. So it felt just, and all these living systems are loops. We get stuck in these views of linear causality, but the living world is full of loops. And so it felt right to be part of this cycle of knowledge and information, and into a page, into words, back into fungus, into human body. So that was it was great fun. And so it's something that people can do. I mean, I find fermentation is a really helpful way to think about these vast biogeochemical cycles which are taking place all around us. The whole planet is enveloped in these great cycles: carbon cycles, nitrogen cycles, phosphorus cycles, and so. But it's very easy. It's very easy for us to forget them because they take place on such big time scales and over such big space scales as well. So fermentation, just in a jar in your kitchen, you can have a fully fledged microbial ecology taking place with waves of succession as one type of microbe gives way to another. And you can taste it as it's happening. You can use your very own mouth to taste how the chemical composition is changing. You have these astonishing flavors that form and they change into another one. The acidity rises, again, it changes. And so you can use your own mouth to, and your own taste worlds to listen to how this biogeochemical transformation takes place. And I think it makes a very good analogy for these much bigger cycles that we too often forget. So a very easy one is beetroot kvass, which is a wonderful drink where you just cube beetroots and you put them wash them, cube them, put them in a jar, and add a 1.5% brine to these beetroots, and let them soak for between 2 and 4 weeks. And you end up with this very life-giving drink, all this sort of rich earthiness of beetroot, but with this acidity from the lacto ferment. It's very healthy and can be used. You can mix it with vodka for a kind of Bloody Mary. You can drink it by itself. It's a staple for me. So that's one thing I'd suggest.
Well, thank you, Merlin. That's wonderful, because that also extends actually our engadinarttalks from 24 hours to 4 to 6 weeks. Because if we now all start today to do this beetroot experiment, we can then, test it and enjoy it 4 to 6 weeks later. So as of today, the engadinarttalks are a 4 to 6 weeks project. So we are very grateful. Maybe a few last questions. I'm interested also in these levels, which are these loads of levels which you describe in the book. In terms of the evolutionary stages, in a way, because you basically say that our evolution has happened in the context of fungi. It's a fungal context our evolution took place in. And there is a biochemical dimension to it, a biochemical process. There is a physiological dimension of it, because the fungi are part of our microbiome. There is a symbiotic level as well. Can you talk a little bit about this? I think it would be great for our listeners to have you explain these different levels and, you know, understand how actually our evolution is so deeply connected to fungi.
Mm-hmm. Yeah, I mean, in general, we think of evolution of different species in relation to their environment, you know, but we forget that or sometimes can forget that their environment is made up of lots of other organisms, you know, so we're always living alongside other organisms. And so the bacteria that live in your gut, their environment is you. And so you and the other microbes that live in your gut. So everything is co-evolving with other organisms all the time. And these relationships that span millions of years leave such an astonishing imprints. We see it very much with plants, with flowers and pollinating insects, and these highly matched forms. So you have the orchid bees, for example, which have evolved to look like sexually receptive female bees, and they attract male bees, and the male bee comes and copulates with a flower, and then goes and copulates with another flower, and in doing so, it spreads its pollen. So there are even species of species of bee that we know once existed but are now extinct. And we know they existed because we can see some specific species of orchid bee which look like these bees. Their biological fidelity, the anatomical fidelity, is so high that we can actually see these bees that once existed but no longer do. So these are very graphic cases, but the same happens with us. And so fungi have imprinted themselves on us in all sorts of ways. And yeasts are a good example. And you think about the beginning of agriculture, a sort of process rather than a strict moment. But yeasts play a very important part in alcohol production and also in making bread. Just to name a few, of course, cheese, many other ferments are key. But if you think about this, say, bread and alcohol, in both cases, humans feed the yeast before they feed themselves. And people often wonder, what was so good about agriculture that caused humans to give up their hunter-gatherer lifestyles, to settle into these new ways of social organization, which brought with them big costs, new diseases, vulnerability to epidemics, ability to accumulate wealth, social inequality. There are lots of questions about why we went for this, new way of life. And one way to think about it is that we were lured into it by these rewards that yeast could provide us with. You know, if we have fields of grain, then it's easier to make beer or bread. The bread before beer hypothesis, or the beer before bread hypothesis. But the beer before bread hypothesis has been gaining traction among scholars for the last couple of decades. But in either case, it doesn't matter which for this point, but in either case, you can think about yeasts in some way domesticating us, just as we domesticate yeast. You know, these arrows always point in two directions at once. So all of these restructurings in human society and in human history happened alongside yeast and the great transformational power of these fungal organisms. And so there are other examples like that too, but there's no organism on living on land that has evolved independently from fungi. And so you can see these fungal habits and lifestyles imprinted in these other non-fungal organisms just by having evolved in the same planet as them.
There could not be a better conclusion, really. I do have a last question, which is the only recurrent question in all my conversation, and it's the question about the unrealized projects. I've always been fascinated that we know a lot about architects' unrealized projects because architects publish them, but we know very little about scientists, visual artists, poets, and so on, unrealized projects. And I mean, the range of the unrealized project is very wide. There can be projects which are too big to be realized, or projects which are too small to be realized. There can be projects which are simply forgotten in a locker. There are also projects one might not have had the time to do them because They're too time intense. There are utopic projects, unrealizable projects, dreams. Then, of course, there are the censored projects. And as Doris Lessing always told me, there is also the projects maybe one hasn't dared to do. She called those the self-censor projects. So there's many possibilities why a project is unrealized. And I wondered if you could tell us maybe about one of your unrealized projects, particularly dear to you.
Such a good question. Well, one of them is an unrealized, not because I think it can't happen, but just because I haven't got around to doing it yet, or haven't met the people who can help it happen yet. But one of the projects is to encourage, fungi have these amazing appetites. They can digest all sorts of chemicals, which are often pollutants or things that we put into the environment and are causing various ecological problems, whether it be crude oil from oil spills or other poisons or heavy metals. So one thing I wanted to try and arrange is a kind of race where people come out of the woodwork from all different parts of the world, where they've been breeding fungal strains that are able to digest terrible poison toxins, and that it would be an event a bit like a cross between a Formula One race and a dog show. And people would come with their different fungal strains, and they would compete to race the fungi through these cocktails of toxic waste to compete for an award of $1 million. And I think this would be enormous fun. I think it would be a great press event and would turn lots of people's minds towards these amazing chemical processes, which we don't think enough about. And also would allow us to discover strains that people had bred, and encourage people to breed new strains of fungi that could digest various types of pollutants. So we'd have some real benefits from it, but I think it would just be a wonderful festival of microbial power, of microbial superpower, and I think it would be really funny as well. So that's one